Tools of recombinant DNA technology. Restriction enzymes. Restriction enzymes belong to a class of enzymes called as nucleases. These enzymes can cut the DNA double helix at specific sequences. These are of two types: exonucleases, endonucleases. Exonucleases. These enzymes remove nucleotides from the ends of the DNA. Endonucleases. These make the cuts at specific position with the DNA. These enzymes recognize specific sequences called palindromic nucleotide sequences in the DNA. Palindromic nucleotide sequences. It is the sequence of the base pairs that read same on the strands when orientation of reading is kept the same. The following sequences read same on the two strands in 5' to 3' direction or 3' to 5' direction. 5' GAATTC, 3' 3' CTTAAG, 5'. The first restriction endonuclease was Hind 2. Cloning vectors. A DNA molecule that carries foreign DNA into a host cell replicates inside a bacterial cell and produces many copies of itself, and the foreign DNA is called cloning vector. Cloning vectors are the next important tools to restriction enzymes. Common cloning vectors are plasmids and bacteriophages. Following features are required to facilitate cloning into a vector. Origin of replication. This is a sequence from where replication starts, and any piece of DNA, when linked to this sequence, can be made to replicate in the next cells. Selectable markers. In addition to ORI, the vector required a selectable marker which helps in identifying the transformants and non-transformants. For example, genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, etc. are used as selectable markers. Transformation. It is a process by which a DNA piece is introduced in a host bacterium. Cloning vectors. These are the sites at which the foreign DNA is combined. Insertional inactivation. Selection of recombinants using antibiotic resistance genes is a complex process. Therefore, an alternate method is developed. In this, the transformants, recombinants and non-recombinants are identified on the basis of their ability to produce color in the presence of a substrate. In this, a recombinant DNA is inserted in the sequence of the enzyme agalactosidase. This results in inactivation of the enzyme and called as insertional inactivation, so no blue colors will be given by this type of bacteria. This indicates the recombination. Non-recombinants will give the blue color. Vectors for cloning genes in plants and animals. Some bacteria and viruses can be used as vectors for transforming genes. For example, Agrobacterium tumefaciens, a pathogen on several dicots, deliver a piece of DNA called tDNA and transform the plant cells into tumor and direct the tumor cell to produce the chemical required by the pathogen. The triplasmid from this bacterium now can be used to deliver genes of our interest into variety of plants. In animal, retroviruses can deliver their DNA into host cells to transform them into concerned cells. So, the retroviruses can be used as vector if the harmful DNA can be replaced by the genes of interest.
In this tutorial, we are going to discuss about ligases joining enzyme in recombinant DNA technology. Ligases are enzymes that are capable of joining two DNA molecules. You can see this is a function of the ligase that is joining two different DNA molecules, and the process is called as ligation. The bond formed is the phosphodiester bond, the bond that is between two adjacent nucleotides. It is between the 5' phosphate of a nucleotide of one DNA fragment and 3' end of the other DNA fragment, and requires energy supply as ATP or NAD+, and an example is T4 ligase. Now let us look into the bond in order to understand what ligases do. And this is a picture showing how nucleotides are arranged in a neat DNA. These are the nucleotides, and nucleotides are joined by phosphodiester bond. And why this bond is called as phosphodiester? And this is the first reason. You can see that from the 5' end there is a carbon oxygen bond that makes the ester bond. This is the first ester bond, and this is at the 3' end of the adjacent nucleotide. This is the second CO bond or ester bond that makes the phosphodiester bond. The function of ligase is making this phosphodiester bond between two DNA fragments. And this is another picture that will tell us the story. This is a missing phosphodiester bond at a 3' end, and what the ligases do is it will synthesize the phosphodiester bond between these two DNA fragments.
Our gene of interest. Gene of interest is isolated from the DNA of donor cell. And then this is our gene of transmit that is also isolated but from the bacteria.
The second process is clean contact. The plasmid and the gene of interest will be cut at the restriction site using the restriction enzyme. They have two enzymes are involved in this process. The first one is EcoR1 that will produce the sticky end, and another one is small one that will produce the blunt end. Okay, this is the example of gene of interest that have cut as the sticky end. And this is the example of plasmid that have cut at the blunt end.
And now we move to the third step, which is insertion. The insertion is the gene of interest, which is a DNA fragment taken from the DNA of donor cell inserted into the plasmid. DNA fragment which is and the plasmid is joined together by DNA ligase, which will form the recombinant DNA.
For the next step, transformation is a process where the recombinant DNA will be transferred into the host cell, such as PAR80 and PR. Their characteristic is able to replicate freely and then able to accept foreign DNA in the MCS, which is multiple cloning site. And then the next one is amplification. The recombinant DNA will be replicated in the cell.
So the last step in gene cloning is screening. So the function of screening is to identify the bacteria that have been transformed with the recombinant plasmid. The bacteria is strict in the Petri plate, which contain H-gal and ampicillin. Only those bacteria contain plasmid with ampicillin resistance will grow and multiply to form a colony. In contrast, bacteria that have not contained plasmid with ampicillin resistance will be killed by ampicillin. Plasmid containing bacteria cell will multiply and form a colony of genetically identical cells. Bacteria with functional beta-galactosidase enzyme will form a blue colonies when they grow in a medium with the presence of H-gal, while if they don't, they will form a colorless colony. Bacteria which form the colorless colony is the one that containing recombinant plasmid and can be isolated for further cloning.
That's all from us.